Hello everyone, and welcome to the INFLANET MOOC course. I am Saatvik Potluri, a Marie Curie Research Fellow at CNRS, and in this MOOC course I will talk about intravital imaging in embryos and how they are performed along with the advantages of different setups. Imaging in models is crucial to understand biological mechanisms and characteristics. Using animal models, we could study different scientific hypotheses clearly and look into cellular interactions and study them at a higher resolution. Models include in vitro and in vivo models, in vitro being cells or tissues cultured outside the organism, allowing us to image them either with bright fields or fluorescence microscopy techniques, and in vivo models include imaging in the live animals like zebrafish or mouse. Imaging in embryos allows us to look at the development of organisms over time and different biological events. Imaging in zebrafish embryos provides us a great advantage, primarily as zebrafish embryos are transparent. This provides us with the opportunity to image events inside the zebrafish over time. It is also easy to build transgenics in zebrafish and label specific cells or cellular components with the help of transgenics. Along with this, zebrafish are well studied and closely related to humans, and studies using drugs could easily be performed in them. To perform any imaging, we must first ask ourselves, what is required of the sample and what kind of imaging are we performing? The setup needs to be optimized for imaging over time or long periods, or if we are imaging at a single time step. The requirements of the sample also need to be considered if it requires temperature control, oxygen conditions, if the sample needs to be anesthetized or immobilized, and if it needs water for the duration of the imaging. This would depend on the microscopy setup being used and if the microscope is upright or inverted. The fluorescence of the sample and what you are imaging also needs to be considered during this. With the setup used, we could image multiple components with different fluorophores simultaneously, but it is important to use emission wavelengths which do not overlap to image them separately. For example, GFP and M-Cherry are commonly used together as their emission wavelengths do not overlap. The microscopes should also be capable of detecting different fluorophores at the same time with multiple detectors and channels. We should also take into consideration if the image needs to be taken in depth or Z to reconstruct the image in 3D. Once this is considered, the time taken to image changes with the use of different setups, and it needs to be checked if this is optimal to see the expected event or if a time lapse needs to be taken to observe this event on the image. The image could be later rendered in 3D to understand it clearer and observe this better. If multiple samples need to be imaged simultaneously, a high-throughput imaging setup should be chosen in which samples are mounted similarly and separate samples are imaged at the same time to lower the variability in the results and get a better data set. The mounting of the sample is crucial to prevent artifacts, reduce variability, and take an image with high resolution. Once the microscope is chosen based on the previous characteristics, the mounting needs to be thought of. Here, in this mounting video, a zebrafish larva at 2 days post-fertilization is placed in a Petri dish with an agar well that is stained with Midland blue to provide contrast for us to see. And the zebrafish larva is placed inside the well, oriented the side that we want to label. And once the zebrafish is well rested in the area, it has also been anesthetized so it doesn't move for long periods of time, allowing for imaging for prolonged periods. After the zebrafish larva has been well mounted, we use low melting agarose that has been stained here with phenol red, and we place the low melting agarose on top of the zebrafish larva to prevent it from further movement and to limit development in a singular direction as well. The zebrafish larva is now stuck in this position, which allows us to image it with high resolution. The reason we use agarose gel is because its refractive index is similar to that of water or PBS, which limits the artifacts it can cause. The zebrafish larva can stay like this for up to 24 hours in this mounting style, and this can easily be used to image the trunk, the retina or the eye region, the cardiac region, or the tail fin region as well. In normal conditions, the methylene blue and phenol red are not used within the agar, meaning they are completely transparent. The transparency of these agar components, as well as the embryo or the larva being completely transparent with very little pigmentation in normal conditions, or using mutated lines which do not have pigments, allow us to image the embryo within depth. And this lack of pigment also limits artifacts or any abstract artifacts in the sample. It depends on if the required resolution could be reached using the specific objective, which could be immersed in the sample container, which might contain solution or not based on the objective. Secondly, the mounting style on if the sample needs to be recovered needs to be very gentle to prevent any damage to the sample and interfere with the event. The mounting style could also damage the sample during the acquisition and shouldn't put any pressure on the sample to observe the event in a stable manner without any external influence. Once the setup has been chosen, it is important to understand the limitations of your setup before making any hypotheses and understanding the event very clearly. Each channel or wavelength is different to one another, as only based on the wavelength can the depth of the image and resolution be determined as previously discussed. Due to this, it is crucial to have a control prior with your imaging experiment to understand if you could actually image the sample at that depth. If a greater depth is required, you could try to use a different microscopy technique like multiphoton microscopy to understand if it might be better for your sample and the event you're observing. Although multiphoton microscopy has its own disadvantages with the need to validate different fluorophores separately and understand their excitation wavelength under that setup. Speed might also be required with your imaging event, especially if you're looking at immune cell movements, calcium signaling, or neural signaling. For this, there are advantages in light sheet and the spinning disk microscopy.